ஆரம்பிக்கலாங்களா?
Demonstration of stomata on a leaf peel. In a plant, the epidermal layer is the outermost layer which contains tiny pores known as stomata. Stomata are found on all above ground parts of plants, including the petals of flowers, petioles, soft herbaceous stems and leaves. Stomata consist of two specialized cells called guard cells that surround a tiny pore called stoma. Their main function is to allow gases such as carbon dioxide, water vapor, and oxygen to move rapidly in and out of the leaf. In different plants, distribution, number, size, and type of stomata vary. In general, the stomata are fewer in the number on the upper surface as compared to the lower surface of the leaf. To do the experiment, we require needle, brush, glycerine, cover slip, a fresh leaf of balsam, glass slide, watch glasses, distilled water, saffron in solution, forceps, filter paper, compound microscope, and a dropper. Procedure: pluck a fresh leaf from a balsam plant. Fold the leaf and carefully tear along the bruised area on the lower side of the leaf. You can see a colorless narrow border along the torn edge. Carefully pull out a thin membranous transparent layer from the lower epidermis using a forceps. Put the epidermis into the watch glass containing distilled water. Take a few drops of saffron in solution using a dropper and put it into another watch glass. Using a brush, transfer the epidermis into the watch glass containing saffron in solution. Keep the epidermis for 30 seconds in saffron in solution so the peel can become stained. To remove excess of stain sticking to the peel, transfer it to the watch glass containing water. Transfer the peel to a clean glass slide using the brush. Take a few drops of glycerine using a dropper and put it on the peel. Using a needle, put a cover slip over the epidermis gently. Drain out excess glycerine using a blotting paper. Take the glass slide and place it under the compound microscope. Examine the slide under the compound microscope. Observations: the epidermis is made of uniseriate layers of cells which have distinct cell walls, a nucleus and cytoplasm, and are closely packed together. The epidermis layers are broken in places. These openings are called the stomata. Each stoma is guarded by a pair of bean-shaped cells which are guard cells.
Dry cells. A dry cell is a modification of wet Leclanche cell. Dry cell batteries are different from wet cell because the electrolytes are contained in a low moisture paste. A dry cell consists of a zinc container whose base acts as the negative electrode or anode. The carbon rod placed at the center with a brass cap acts as the positive electrode or cathode. This is surrounded by a mixture of manganese dioxide and charcoal in a muslin bag. The electrolyte used is a moist paste of ammonium chloride. The outer body, excluding the base of the zinc container, is insulated with cardboard to prevent graphite rod from coming in contact with zinc container. Working of a dry cell. When the dry cell is connected in a circuit comprising a bulb, current flows in the circuit due to the chemical reaction that takes place in the cell. This makes the bulb glow. The electrode in the dry cell battery has two terminals, one that is positive and one that is negative. When a bulb is connected to the battery's terminals, a chemical reaction occurs between the electrode and the paste that produces roughly 1.5 volts of electricity. The electricity produced is conducted out of the battery with the help of an external circuit.